Hello everyone, welcome back to my channel Capsa. Have you ever wondered how some bacteria can lie dormant for hundreds of years unaffected by boiling water, radiation, or chemicals? The answer lies in the most powerful survival tool, the bacterial endospore. In this lecture, we are going beyond the structure. We will explore the biochemical secrets that give these spores their unmatched resistance and then look at how they return to life through a process called germination. Let's start with the question: what makes endospores so resistant? The answer is not one thing. It's a multi layered defense system, both structural and molecular. The first key player is dipicolinic acid or dipicolinic acid, often abbreviated as DPA. This compound, in association with calcium ions, accumulates in the spore core and can make up nearly 10% of its dry weight. These molecules stabilize proteins and nucleic acids and importantly reduce the water content in the core. Now that brings us to the dehydrate core, another critical factor. With water content as low as 10%, there is almost no free water for enzymes to function or for hydrolytic damage to occur. This dehydrated state puts metabolism on pause and preserves macromolecules in their inactive, stable forms. But bacteria do not stop there. They produce small acid soluble spore proteins or SASPs. These special proteins bind tightly to the DNA, protecting it from the UV radiation, desiccation, and enzymatic damage. They also compact the DNA into a more resistant configuration. Surrounding the core is the cortex, made of a specialized peptidoglycan that maintains core dehydration and assists in spore dormancy. Outside the cortex, the spore coat, made of protein rich keratin like material, provides resistance to chemical and enzymatic attack. It's highly cross-linked, forming an impenetrable barrier. And finally, hidden within the core are the DNA repair enzymes. So these remain inactive during the dormancy but into action bring into action as soon as germination begins, quickly repairing any accumulated DNA damage before the cell resumes the growth. Now let's switch the gears. What happens when conditions improve? Endospores don't just passively wait, they are equipped with germinant receptors located in their inner membrane. These receptors can detect environmental signals like amino acids, sugars, or low pH, clues that the external world is safe again. Germination is a three-stage process. First comes the activation. This is usually induced by a mild heat shock or specific chemical exposure, such as alanine or inosine. It's like warming the engine of a long unused car. So next is true germination. Here the internal changes begin. The spore releases its dipicolinic acid and calcium ions and begins to absorb the water. Enzymes become active and the cortex is degraded and the core rehydrates. Finally, we reach the outgrowth phase. At this point, the dormant spore becomes metabolically active. It begins synthesizing proteins, DNA, RNA, ruptures the spore coat, and emerges as a fully functional vegetative bacterium. So, this remarkable transformation from a dormant stone like particle to a living, replicating cell can occur in minutes once triggered. So, what wakes a spore up? Nutrient availability is the most common signal, especially amino acids like alanine, L-alanine. A mild heat shock can also prime the spore to germinate. In this case, pathogenic spore formers, the presence of host tissues or gut bile acids may initiate the germination process, turning an environmental spore into a disease causing cell. Endospore resistance presents significant challenges in medical and industrial settings. In hospital, routine disinfectants often fail to kill spores. This makes pathogens like Clostridium difficile a major cause of hospital-acquired infections, especially after antibiotic use. To ensure sterility, we use autoclaving, which applies saturated steam at 121 degrees Celsius under pressure for at least 15 to 20 minutes. Even then, to validate this process, we use biological indicators, usually spores of Geobacillus stereothermophilus, because if these die, we can be confident everything else is dead too. In the food industry, survival leads to spoilage and foodborne illness unless adequate thermal processing is ensured. Even in spacecraft assembly, spores are a concern as they may contaminate other planets during exploration missions. The resilience of bacterial spores has been weaponized. For example, anthrax spores were used in bioterrorism attacks in early 2000s, sent through the mail. These spores can remain viable in soil for decades, waiting for the chance to infect a host. Understanding germination has therapeutic potential. If you can block germination within the host, we might prevent disease caused by C. difficile or C. botulinum before toxins are even produced. Thus, the study of spores is not just academic, it touches on public health, infection control, industrial microbiology, and even planetary protections. So, there we have it, the biochemical secrets behind the resistance of endospores and fascinating process through which they return to life from dormancy to reawakening. Endospores showcase one of the nature's most effective survival strategies. So, if this series helped deepen your understanding on microbiology, considering giving a video thumbs up, sharing it with your classmates, and subscribing to the channel. And don't forget, science never sleeps, and neither should your curiosity. See you in the next lesson.